Hello and welcome back. In the previous session, I put forward a question. How do the neutrophils exert their functions in the inflamed site? You will get the answer today after I introduce leukocyte functions in inflamed area. When we finish this session, you should be able to answer what functions do leukocytes exert in inflamed area, and what are the steps involved in the process of leukocytic phagocytosis. After transmigration, leukocytes continue to move towards the site of infection or tissue injury by chemotaxis. Once recruited to the inflamed area, leukocytes are activated to perform their functions by using various receptors to sense the presence of noxious stimuli and cytokines. For instance, G-protein coupled receptors, toll-like receptor, cytokine receptor, or phagocytic receptor may recognize different stimuli to initiate cellular responses that mediate leukocyte functions. Leukocyte activation results in the enhancement of the following functions: phagocytosis, intracellular destruction of phagocytosed microbes and dead cells, release of substances that destroy extracellular microbes and dead tissues, and production of inflammatory mediators that amplify the reaction by recruiting and activating more leukocytes. Therefore, while the enhancement of these functions would facilitate destruction of noxious stimuli, amplify the inflammatory reaction, they would also cause tissue injury at the same time. Generally speaking, the main functions of leukocytes in inflamed area involve phagocytosis, immune response to microbial or foreign antigens, and leukocyte-induced tissue injury. Let me introduce them one by one. Phagocytosis is an important defensive function of the body. It embraces the ingestion process of particular material by leukocytes. Neutrophils and macrophages are the most important phagocytes. Circulating monocytes enter the tissue and then differentiate into macrophages. Phagocytosis involves three sequential steps, namely recognition and attachment, engulfment, killing and degradation of the ingested material. These steps are triggered by activation of phagocytes by microbes, necrotic debris, foreign bodies, and various mediators. The first step of phagocytosis is recognition and attachment. This step depends on specific surface receptors of the phagocytes. There are two ways for phagocytes to recognize and bind particles. One is recognition by opsilin receptors. The other is by non-opsilin receptors. What are the opsilins? They are host proteins that enhance the efficiency of phagocytosis by coating microbes and targeting them for phagocytosis. Immunoglobulin G antibodies, complement C3b, and certain plasma lectins are the major opsilins. The process of coating microbes by opsilins is called opsalization. This picture shows this microbe has been coated by IgG and C3b. Phagocytes express receptors for opsilins, such as the Fc receptor for IgG, complement receptor for C3b, and C1q for the plasma lectin. Recognition and binding of opsalized microbes facilitate their rapid phagocytosis. Non-opsilin receptors, such as mannose receptors and scavenger receptors, can recognize and bind components of microbial cell walls. The second step is engulfment of particles. Look at this picture. An oxalized microbe is attached to the phagocyte surface by binding to the corresponding opsilin receptors. Then the phagocyte extends pseudopods to enclose the microbe, forming a phagosome. The phagosome fuses with cytoplasmic lysosomes and forms the phagolysosome, into which the lysosomal contents are discharged. Phagocyte kills and degrades the ingested microbe in the phagolysosome. The final step is killing and degradation of the ingested material. It is accomplished within the phagolysosome by oxygen and nitrogen dependent and oxygen and nitrogen independent killing mechanisms. Oxygen and nitrogen dependent mechanism is the most important intracellular microbicidal mechanism, stimulating an oxidative burst with a rapid increase in oxygen consumption. This mechanism mainly depends on the production of the reactive oxygen species, ROS, and reactive nitrogen species. Among them, hyperchlorous radical is a potent oxidant and antimicrobial agent. Oxygen and nitrogen independent mechanism is performed by proteins such as lysozyme, lactoferrin, major basic protein of the eosinophils, bactericidal permeability increasing protein, and defensins. Lastly, the dead microorganisms are degraded by the action of lysosomal acid hydrolases. How are ROS produced? The generation of ROS is due to rapid activation of leukocyte NADPH oxidase. Look at this picture. NADPH oxidase reduces oxygen to the superoxide anion, which is further converted into hydrogen peroxide. Hydrogen peroxide is not able to kill microbes efficiently by itself. However, the neutrophil lysosomes contain the enzyme myeloperoxidase, MPO, which, in the presence of a halide, converts hydrogen peroxide to hyperchlorous radical. The latter is a powerful oxidant and antimicrobial agent. Therefore, the hydrogen peroxide myeloperoxidase halide system is the most efficient bactericidal system in the neutrophils. Besides phagocytosis, leukocytes also exert immune response to microbes or foreign antigens. This function is mainly performed by macrophages and lymphocytes, which mediate cellular immunity or humoral immunity. Leukocytes also exude contents of lysosomal granules outside the cells, such as enzymes and ROS, to destroy and digest extracellular microbes and dead tissues. However, this also damages normal cells and tissues, exacerbating the harm caused by initial inflammatory stimuli. Therefore, besides exerting defensive effects, leukocytes can simultaneously induce tissue damage. Now, I think you should be able to answer the question, how do the neutrophils exert their functions in the inflamed site? Let's make a summary. Once recruited to the inflamed area, leukocytes are activated by recognizing microbes, dead cells, and inflammatory mediators. Activated leukocytes exert phagocytosis, induce immune response, and produce inflammatory mediators to amplify the inflammatory reaction. Leukocytes eliminate noxious stimuli, mainly through phagocytosis, as well as via extracellular release of lysosomal contents that would also damage normal tissue at the same time. We have talked about the process of acute inflammation through 3 sessions. Acute inflammation subsides after the offending agents are removed. If the offending agent lingers, chronic inflammation will develop. Next time, we'll talk about different morphological patterns of inflammation. That's it for this session. Here are the references. See you then.